Okay. So hi, everybody who's here, who's watching. We're gonna go over the first part of the cell cycle. We broke it up because there's a lot of things involved in the cell cycle. Um, so yeah, this is the first part where we're gonna go over like the intro to it and then some G1 stuff. I think on Friday's session, they'll probably go into like S and G2 phase. But next week, you're gonna go over like restriction points and all of that. So this is just the intro to it, but as always, if you have any questions, you can email either myself or Chi and or any of the other TAs, and we'll get back to you. Okay, so this is just an overview of everything in the cell cycle. So let me put a little pointer on. So we start with this G, or I guess G0 is kind of where everything starts, but G1 phase is the start of the cell cycle. The G stands for gap or growth, because we have these two of them. We have G1 and G2, and during both of these phases, the cell is pretty much growing. So during G1, the cell prepares for DNA replication, which will occur in S phase. And in order to do this, we need the cell to have enough nutrients, cell machinery, and no mutations in order to have like smooth DNA replication. This is where the cell does commit to the cell cycle. And there's an end of phase checkpoint, as all of the phases have them, that you'll notice. So S phase has an end of phase checkpoint and G2 phase has an end of phase checkpoint, just to make sure, because we can't have the cell be entering different phases without making sure everything's okay. And you'll learn more detail about that, I think, next week. So this is G1. It's the longest one, pretty much. There's this G0 phase, which is the non-dividing state. This is in cells that just don't divide. They just won't enter the cell cycle at all, because they don't need to divide. So once we move through G1 and we make sure the cell has grown enough, that there's enough nutrients and all of that, and we pass the checkpoint, the cell has now committed to the cell cycle, and it can't leave it after this point. And then we enter S phase, which S stands for synthesis. This is where DNA replication occurs. Once we pass that checkpoint, DNA replication went well. Then we are entering G2. This is the second gap or growth phase. This is where we prepare for mitosis to occur, or chromosome segregation. So more growing will occur, the spindle assembly and all that forms, and I think it's M phase actually. But there's another checkpoint, and then we enter M phase, which is we go through prophase, metaphase, anaphase, telophase, cytokinesis, all of that, because this is where the cell splits into two. And then we can start it all over again if the cell needs to divide again.
Okay, so now CDKs, or cyclin-dependent kinases, are kind of like the cell cycle regulators. They regulate everything that happens in this cycle. They're mostly like serine and threonine kinases. So like they require these protein complexes for everything to occur. And for CDKs, they are also regulated by cyclin, but that doesn't really mean phosphorylation or dysphosphorylation doesn't mean it's active. It needs another factor for it to be active. So for CDKs, it needs to be actively being phosphorylated, and also for the cyclin to be bound to it as well for it to be completely active, as like the figure on the right shows, needs both phosphorylation and the cyclin bound to it for it to be active as well. And then once you reach a checkpoint, like you talked about in a previous slide, the cell kind of puts like an inhibiting or deactivating CDK, which are kind of like the brakes to kind of pump the brakes and make sure everything is working correctly and stuff.
Anyone have any questions so far? No. Okay.
I'm good. Thank you.
Yeah, so like we mentioned, cyclins are required for CDK kinase activity. They activate CDK's kinase activity and also tell CDK what to phosphorylate. So you'll notice throughout like future lectures and next week's material that there's like M cyclin, there's like S cyclin. They're specific to certain things of what they need to what CDKs need to phosphorylate. The cyclin regulation is involve a transcription and translation at specific points that they're needed in the cell cycle. This is how they're formed when they're needed. And then when cyclins are not needed, they get tagged with ubiquitin, and then they're sent to the proteasome for degradation. This is ubiquitin-mediated proteolysis. And this is just to get rid of extra cyclins that aren't needed. But if you see here, you can see the different cyclins that are involved. So the levels of cyclin D1 during G1 phase increase, and then they kind of drop off around here. And then cyclin E kind of increases here, cyclin A. You don't need to know these specific ones, but this just shows you different types of cyclins and how they're kind of increased and decreased wherever they're needed during certain parts of the cell cycle.
So this is an example of how.
Sorry, I have a tiny question. So what is the use of the inhibitory CDK again? I remember you said it. I just didn't write it down. Sorry.
No, that's okay. So like what's the use of an inhibitory CDK?
Yeah.
Yeah, so we're actually going to talk about that in a second. Yeah, it's basically, no, you're fine. It's basically just like if the cell, so like, you're going through the different phases and there's the checkpoints. If the cell, like say you're replicating DNA and there's DNA damage, then you need to stop, like you need to put a break on the cycle, because we don't want like mutations to accumulate, because you want to make sure everything's okay for the cell to divide.
Okay. Okay, yeah. I can actually see in the screen. Thank you, I appreciate it.
Yeah, no worries.
So that's actually what the screen, what Rachel talked about, what this particular slide is talking about. So like an example, if there happens to be a mutation right after G1, usually after each phase, the cell, polymer cycle break just to make sure everything's working correctly. So now if there's like a mutation in G1, P53 would get activated. And P53 is a transcription factor, and it's also a tumor suppressor. So then that's what responds to DNA damage if there is one found. And if there's DNA damage found, then P53 activates P21, which inhibits CDK. So inhibiting CDK will kind of like put a break on the cell cycle. So like nothing will go forward, like the cycle will stop for a minute to try to fix the issue. Now, if the damage can be fixed, then it gets fixed. But if not, then P53 can signal for apoptosis, which is just that particular one, just getting through ubiquitin proteolysis that we talked about on the previous slide. And P53, once like it's active, once like it's needed, it's produced like in heavy amounts. But if it's not, then it gets degraded as well, so it doesn't interfere with anything else.
Yeah, so this is just like Chichi said, one of those examples as to why we would need CDK to be inhibited, because we don't always want the cell cycle to be going if things aren't right, if that makes sense. Like, you don't want to divide cells that have like a bunch of mutations and lead to problems down the road. And this is why P53, one of these tumor suppressors, this is why cancer can get out of control, because you have rapidly dividing cells with no breaks involved. And this is why you get tumor cells and proliferation of cells, because they don't have a signal to keep them just like to stop dividing, basically. So we want inhibitory CDK. Okay. So now we're going to G1 phase. So this is that first phase, that kind of longer phase. And this is where the phase where cells have to commit to the cell cycle. So before cells do enter the cell cycle, they have to receive growth factor signals. They have to check for DNA damage. They have to check for cyclin D, which is one of the early G1 cyclins. They have to check for size. This is why it's called a growth phase, because we need to increase the cell size. They need to have enough nutrients and energy, and they need to make sure that the pre-replication complex is formed, which we'll talk about in a second.
You want to just continue?
Yeah, I can keep doing it because that wasn't really like a slide. Okay, so this pre-replication complex, or the pre-RC, forms when activated. So it begins to form when activated CDK levels are low in G1. This is because CDK inhibits the formation of the pre-RC throughout the rest of the cell cycle. The reason for this is we only want one round of DNA replication, because you don't want to replicate your DNA multiple times. You only want to replicate it once. So this is why the CDK inhibits the formation of the pre-RC throughout the rest of the cell cycle. So it starts to form at the beginning when CDK levels are low. So we do have a ton of origins of replication along our DNA. So this blue thing is the ORC complex, or origin of replication. There's a bunch of them along the DNA. We're just focusing on this one in particular. So basically what happens to form the pre-RC is the ORC binds to the origin site. ORC is an ATPase, that means it hydrolyzes ATP hydrolyzes ATP, sorry. ORC recruits the CDC6, which is another ATPase. So you get the CDC6 to bind to ORC, and then when CDC6 is bound, then CDT1 can bind because CDC6 recruits CDT1. Just a side note, to help you remember these, I like to draw them out, or it just helps me remember the order of things, because I know reading these out loud won't really stick for a lot of people. But so you have the ORC, then CDC6, then CDT1. CDT1 recruits this MCM complex. So MCM is a helicase, and I'm sure you all know what helicases are. This 2 through 7 is just like what the subunits are named. But anyway, so MCM, so now you have this whole complex, which is the pre-RC. But one thing to keep in mind is once CDT1 recruits MCM, it dissociates. So these proteins all together on the DNA form the pre-RC, or the pre-replication complex. This only forms when CDK levels are low, because CDK levels will inhibit it throughout the rest of the cell cycle, because we only want replication of our DNA to occur once. So this is all set up so that when we go into S phase, we can use this to replicate our DNA. That's why it's called the pre-replication complex. Does anyone have any questions? No? Okay.
I have a stupid, it may be a stupid question.
No.
But like, okay, so the role of the CDK is to help the cell divide, right?
Mhm.
So is there like a chemical reason why a pre replication complex is formed that is used for replication, and like CDK is used for cell division? You know what I mean? I don't know.
Yeah, no, I know what you mean. I think it's just that the CDK levels kind of regulate when things happen, and then these are like the things that CDK is regulating leads to certain events occurring throughout the cell cycle.
Yeah.
But I don't know for sure like the chemical reason,
Okay.
if that makes sense.
Yeah. Thank you. I appreciate it.
Yeah, no problem. Sorry, I couldn't give like a better
Nice.
answer.
Okay, so like she talked about in G1, CDKs are regulated for in order for the pre-replication complex. So then in S phase, for it to move from G1 into S phase, the CDK activating kinase will add an activating phosphate, but then V1, which is another nuclear kinase, will add an inhibitory phosphate. Now, if both of those are added, the cyclin CDK will still be inactive because one is activating it, but the other is inhibiting it. So in order for the CDK complex to be active, you need to cleave this inhibitory phosphate. And in the little diagram below, once the CDK is bound to the cyclin, so they become active, but the inhibitory kinase, which is V1, binding to it, as well as the activating kinase that also causes it to still be inactive. So in order for it to be active, V1 needs to be cleaved, which puts the inhibitory phosphate on it.
Yeah, so so based off of that, so what happens is you, like she just said, inhibitory phosphate from WEE1 and the activating phosphate is they're kind of canceling each other out. So this is an inactive CDK. So in order to make it active, like Chichi said, we need to get rid of this inhibitory phosphate. So CDC25 is a phosphatase, so it removes this inhibitory phosphate. So now all we're left with is this activating phosphate. So since there's only one phosphate and it's the activating one, the CDK complex is now active. Does that whole process make sense?
I just had like a question. So like, are the inhibitory and the activating like always added at the same time, or is like the activating like added sometimes like by itself? Does that make sense?
Yeah.
Okay.
I think in this case, because we're in G1, that what happens is they're both added at the same time to give the cell time to like do everything. And then this is how and then this next step where we take off the inhibitory phosphate is how we transition into S phase.
Okay.
So it's there in this particular case, yes, they will be both added at the same time, just because we need to set it up for the transition into S phase. So during that like growth phase and everything happening, pre-RCs being formed, all that, these two need to be added because we want this activating kinase, we want this activating phosphate there. It's just a way of WEE1 making sure we have everything set for the cell. And then when it's like, yep, okay, got the green light, can go ahead. Then CDC25 will just remove this phosphate.
Okay, cool. Thank you.
No problem.
Now, this is the restriction point of G1, which is right before S phase. So now after CDC25 has been added to the cyclin CDK complex, now it's active. Now activated CDK will phosphorylate protein RB. RB is originally bound to E2F, and with RB bound to E2F, E2F cannot do its function in the cell. So now with CDK phosphorylating RB lets go of E2F. And now with E2F let go, it goes into the nucleus. It's a transcription factor. So now once it's in the nucleus, it starts transcription. And then this gets everything ready for S phase. And now once the cell passes this kind of checkpoint, it can't go back. There's no returning, there's no nothing. Now S phase starts. Any questions? Anything? Cool.
Yeah, so after the pre-RC is assembled, so that whole thing with ORC and MCM and all that, other proteins are added to make the pre-initiation complex or the pre-IC. So this complex does require active CDK for formation. So if you remember that the pre-RC can't form with a lot of CDK levels, but the pre-IC needs like more CDK levels to form. So after the pre-IC forms, polymerases can be recruited. And once the polymerase is added, then DNA replication can begin. And this is when we officially enter S phase. So this is the point of no return, this restriction checkpoint. And each cell cycle phase has this restriction checkpoint. It has like a certain one, not the specific one, but they each have their own. But this is kind of like our form, like our initiation to replication. And then this is where we enter S phase. Yeah, so that's pretty much it for G1. And then I think Friday's gonna go over S phase, but this slide was just like some main ideas about the cell cycle. I think we'll include them like with most of the slides because I know it's a lot of information, but just remember that the cell cycle events happen in a particular order to make sure that things go smoothly and that there's no risk for error, mutation, all that. The cell cycle events involve cyclical activation or deactivation of proteins. This is often by means of phosphorylation with ATP. CDK, like we said, must be bound to cyclin in order to be active. CDK regulates particular proteins in different phases of the cell cycle. Different phases have their own cyclins, like cyclin D, M cyclin, all that. Each cyclin is created and degraded in particular parts of the cell cycle. And regulation for proper functioning requires activation / deactivation and inhibition. So it's not so much as more positive or negative, it's just activation or deactivation, depending on what the cell needs. And that's all we have for you guys. Does anyone have any questions?
Sorry, for the really last one. So you said the pre-ice, sorry, the next slide. Yeah, that one. You said the pre-IC needs a lot of CDK, but cannot according to presence of CDK.
So I actually don't know if it needs high levels of CDK. It just requires active CDK for formation. So I'm just assuming that it would need more levels of it to form, but I actually don't know if it's specifically higher levels of CDK. It just requires like active CDK to form. Does that make
Okay,
sense?
thank you. Thank you.
Yeah, no problem. Any other questions?
So this is gonna be counted as our check-ins, right? As our weekly check-ins.
Yeah, since there's only 2 of you, yeah, this will count.
Okay. Plus you're my TA too, so that's
Yeah,
good.
yeah,
Thank you.
I'll write you down, no worries. If you guys don't have any other questions, have a good night and let us know if any questions arise.
Thank you.
Thank you. Thank you.
Bye, guys.